The film today is about a truly amazing discovery of an animal included in amber. Actually, my job is not involved with amber. I'm a gemologist who is testing gemstones and have my network in Burma and all over the world who miners and gemologists and experts who bring me all the news from all over the world, from the mines. Now I will have to focus on the extraordinary experience I made with this research project and the outcome on one particular sample. You find amber everywhere in the world. Colombia, Indonesia, Baltic Sea, and Borneo, Sumatra, and different colors and all kinds of materials. But if you want to find this one here, then you have to go to only one place in the world, in northern Kachin state of Burma. It's not only Burma, because Burma also produces 4, 5, 6 different places, but only one place in the Tiger Valley near Tanai in the northern Kachin state here. So this is exactly this amber from northern Burma. It's called Burmite, with a life form included. This example here is a lizard, a little bit decomposed and the tail is off. This piece is 10 grams and it's 2 centimeters. It contains a lizard that lived together with the dinosaurs 100 million years ago. To finding a lizard in an amber, you need to cut about 1 million pieces of amber. And this is how you want to do that. You need the community work, the work of an entire community to find that species. You cannot go just into a hole in an amber mine and say, okay, this layer, now we're going to find that lizard, and you hammer, you take a bag out, you run out and you cut this stuff and then you find the lizard. No way, it's not going to happen. You're going to never find it. Your whole lifetime, you're never going to find a lizard. Even if you are 10 years yourself digging in a mine, you need 20 to 50,000 people working in 150, 200, 300 mining spots, and they're finding 10 or 50 lizards a year. And somebody has to open this rough material and have to polish it and look inside and see what's inside and find that lizard. And only the Burmese community have enough miners to do that. And that's the reason why the Burmese inclusions in the Burmese amber are so important for the world, and why they have taken the world, because it's the effort of the indigenous people, of the miner, of the hard-working people that found this material. So first of all, I give them the credit. What is my role? My role is to connect to these people.
It is amber.
Amber.
Amber. Yeah.
What we heard from the market was that in Myitkyina, in the Kachin state of Burma, they discovered an amber deposit. And this amber deposit is 100 million years old. Actually, what means 100 million years? At that time, the dinosaurs were still running around. It was in the Cretaceous. So it captured the attention and we started going to looking into this material. What is amber? Amber is part of a tree. It is resin that the tree expels when it gets hurt or cut off something to heal off. And this resin, if it's put under high pressure and a lot of overlaying rocks, transforms into an amber. So it's a total different world than the gemstones. In the gemstone we look at the inclusions of minerals, and in the amber we look of inclusion of animals and plants that were living together with the dinosaurs. This was the heydays of the dinosaurs. There was a huge diversity, and now we have a witness here, like a fingerprint of the environment and what happened there. And once we started to go and collecting more and information came together, and here it was becoming like a huge wealth of information. A whole new ecosystem totally disappeared and now frozen inside this amber material. First of all, we found the plants, leaves, and there were ferns. Next to it were flowers, and with the flowers were butterflies and wasps for pollination of the flowers. And on the ground were snails. In the air were birds. We found their feathers. And then there were hunters, dragonflies that hunted all the flying insects. There were spiders, and there was one sample who had the spider. There is still the net there, and you see what it catched. And there was hair of animals that were running around. And we have beetles, millipedes, so many different flying insects with 2 wings, with 4 wings. And then there was this swarm of mosquitoes. One mosquito even had a little bit blood inside. When I look to this collection of amber, I feel an enormous joy in my heart. It is this unique assemblage of all these pieces with information about a world that disappeared. And now it's here, all collected. Is this opportunity to reach out to all the other scientists that can go and study and make a contribution to the knowledge of evolution of animal on the planet Earth. Okay, let's see. Yeah, okay. One more and
One more.
a nice one. Okay, here it is. Oh, nice! Ahaha. Nice legs and everything. This one can see very well. And so life. Wow. Man, were we hard looking for anything that flies or walks, you know? Here it is, here. It's very beautiful. A very small one.
I small one. I see.
You see it?
There. Yeah.
Here, you see it? It's life, you know? We were looking, look, now we have 19 kilo of amber.
Yeah. And
Work
we
through.
found
We worked up
1.5 milligram of live.
in 19 kilo, kilo of amber. Look, I'm telling you, I just wanted to discover life in amber
Yeah.
myself. We have some more pieces that I can look in, or?
I need to finish the polish.
Okay, good. You want to make the discovery in the world of life in amber, you ending up finding an important piece to explain the history and geology of amber formation. You still make the discovery, but you have to be patient. And then the respect for all the miners, the thousands of people that helped in this project, of cutting 200,000 pieces and almost 1 ton of amber up to finally make a discovery. So I learned, and I'm very humbled now with the miners. How can they find all these things? I respect now their work much more than before I could do it, because I never tried myself. Now I tried myself, now I know it is an unbelievable hard job to find life in amber. At the end of the whole process, and being the guy who pulls everything together, invest into the project, and having the job to make the library and the whole collection as a rescue investment fund for the evolution of the Earth, I'm very humbled and happy that I can do that. And I have the network to the university, so I can go and supply all these many lizards, scorpions, spiders, insects, all these new discoveries. There are hundreds and hundreds. I couldn't talk today about it. And I put them all in the pipeline at the important universities. So we will discover new species, I think at least 15. And we will publish them, we will name them, we will join with the most important scientists of the world, and it's another little lifetime job next to my job as a gemologist. It took 10 years to come where I am right now. We have museum pieces. The big amber will go in a museum, in our own museum, and display. The fluid inclusions will be researched, and we learn more about the paleoclimate, about the paleotectonic processes in amber. So it's an asset. I play the role in rescuing this part for the world, and I did it because of passion, because of a role I can play at the end of my career, pulling this all together. And now comes the big next job to make sure it's not get lost for science, and it makes its way into the world and into the newspapers. So we had to go and make a research tour in the world, and we decided on the advice of Juan Daza, a researcher on herpetology in USA, Houston, Texas, to visit the CT scan laboratory in Austin, Texas. For this purpose, we had to export all our material, and officially from Burma to Switzerland, and from Switzerland to the United States, and then to this absolute fantastic laboratory, UTCT. And it was very fortunate, and we are very grateful that we could go actually and see how they are measuring our samples in this laboratory. So we were welcomed by the two leading scientists, Dr. Maisano and Dr. Colbert, in that laboratory, and could explain and follow up the entire process, which we are now documenting for you.
Well, my name is Jessie Maisano. We are at the University of Texas High Resolution X-ray CT facility, or UTCT. This is a facility that was established more than 20 years ago. I've been here almost 20 years, since 2000, and I manage this facility. And the purpose of the facility is to make industrial strength CT scanning available to academic researchers. The poster just kind of is a nice summary of the variety of things that we scan, everything from the forehand of a prosauropod dinosaur, which you see the picture of the actual fossil here, and then after it was scanned, we were able to digitally go in and isolate each bone of the fingers so that they could be taken apart and described. So this is our ultra high resolution system, and we typically use it for things requiring about 5 micron resolution and higher. So this is the X-ray source. It's 150 kV. It goes down to 40 kV, so for things that don't stop X-rays very well, like feathers and paper, it's good to have that lower energy part of the spectrum. I've been scanning your samples at 70 kV. So the sample sits on the stage, unlike medical CT where you lie still and everything rotates around you. It's the opposite with our systems. So the sample will rotate on the stage while the source and the detector stay stationary. So this is the detector, and this is the camera that captures the data. And this system actually has 6 different detectors on it. This is the lowest resolution, but on this turret we have a 1x, 4x, 10x, 20x, and 40x. And just as it looks, they're mounted on microscope trays. So Zeiss, what they do differently from every other scanner manufacturer is they use geometric magnification. You drive the source as close as possible to increase the resolution. But then after you've done that, as close as you can without hitting it, then you move the detector further away, and that further increases magnification. So you can do small subvolumes of larger samples on this system far more easily than you can on a standard CT scanner. So on this system, the highest magnification we can achieve is a quarter of a micron.
So I was like, okay, we're going like this. We're not going to take a lizard and checking with somebody, and then he says, okay, we need a CT scan. We have CT scan everything, the entire collection, everything that's important, over 100 samples.
Well, you know, I don't want to say it too much because otherwise then you are not discovering it, then I'm discovering it. But I want you to discover it. This one is a piece that I don't want to tell you what it is. It should be. I want to have a surprise momentum. Yeah. It's the piece, like turn it like 90 degrees and tilt it and so on. You can't see it.
So we went there and a whole project through, tack, 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 because they have to measure it, then they have to, this is like 2,000 sections, X-ray scans through the stones, like this through, you know, zoom, 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 and then later they have to combine the information to make a 3-dimensional picture of the inclusion. So it measures hours and days and nights, and then you have to calculate all the information, and then you get a data set. This data sets were hundreds of gigabytes, terabytes, 10 terabytes of information from all the measurements we made. And these 10 terabytes, if you want to interpret it, we had to buy a special workstation at our laboratory so we can even look at this picture and create movies of the lizards and all that. So this was like a huge machinery in a total different world of huge data sets. Weeks of measuring for like 15 samples, weeks. So 100 samples is like, I don't know, 10 weeks measurement, maybe 7. And of course the scientific overhead is very important, but I was realizing how much we forget all the people in the laboratories that really do the work. Oh my God.
Because we have to do a helical scan. So right now here we just went to see.
Okay, so it was very interesting, and the guys were freaking out about the findings. Never see this in my life. They said in this one. And they were realizing, so this is a world-class collection that came out from us. The CT scans at the UTCT laboratory in Austin, Texas, could be complemented by another type of machinery that works differently. A synchrotron facility in Australia. So now we had to travel from Austin, Texas, first to Hong Kong, and from Hong Kong to Melbourne, with all the difficulties of bringing a collection with you, and then visit an unbelievable, stunning laboratory and machinery park in Melbourne at ANSTO. I was very surprised when I saw this huge facility.
So this is a workhorse synchrotron. So it's 3 GeV. So there are many synchrotrons of this type around the world, but it's new. So it's got new beamlines, new magnets. It's very high efficiency. It's in its early days of operation, 10 years old. So I think it's got 9 or 10 operating beamlines. The aim is to have 30. But if you have a look down here, we've got things like synchrotron imaging is bread and butter. And we're here actually studying a number of Cretaceous birds, but we've also got a number of your precious amber samples. And what we're about to put on the beam is this sample, which is a large scorpion. Now the instrument is set at 11 micron pixel size because the sample is relatively large and we don't need the high resolution. But if you come with me, let's go load the sample. Step on the way in. So this is the imaging medical beam line. The radiation source is just over here. So the synchrotron is about 140 meters or so. I have to check the number. But the radiation beam is coming out through here actually, and then passing right through this chamber to our sample stage, where I place the sample, and then we've got the detector here. So bubble wrap's transparent to synchrotron X-rays, so we're just going to place the sample carefully between these carbon fiber poles. So this is just to make sure that there's nobody in the room when the X-rays are operating. So we've got to search every corner of the room. And we need to go through this search process every time we close the room. To the computing hub, where we can see the sample live on the screen.
Having this hundreds of samples of synchrotron data, I established, we realized we have like 20 terabytes of information. And this information we cannot send all these samples and all this information to scientists. We have to bring the scientists to the sample and to the data and make a symposium. So what was the idea? We are going to collect 15 to 20 the best scientists we can find in the world in different fields, entomology, herpetology, and geology, luminescence, photography, spectroscopy, working in this field, and bring them to make an almost 1 week seminar, bring them all together, build a workshop on the table, all this microscope up. Everyone needed, oh, where is my microscope? Oh, where is my light? Or where is my samples? What do we have? Where is this? Where is that? So I have to. My whole crew from GRS Thailand was busy. And my special diamond room was plugged out, emptied, and made a huge seminar room. And there were like 6 to 7 work groups, 3 to 4 people, or 2 to 3 people working together on jobs. One on scorpion, one on lizard. In another room, a whole group on lizards. Then on feathers, on dinosaurs, on all these subjects we had in Amber working as a workshop and interacting together. So then we made the big plan on the blackboard, who found what, where we are going to publish, what we are going to do. We are listed at everything, and here it is, and this one, and this one, and this one. Everything was listed and planned and made, and a huge, nice, like 15 to 20 publications came out. New discovered insects, new lizards, new species, new discoveries, and then happened something very dramatic, very dramatic.
Jesus.
Be careful if it falls unfinished. Only keep it 1 centimeter high.
So gorgeous.
So what are we looking at here? It's obviously a skull of an animal that was found inside amber. And obviously there is no amber here because it's not a picture of the skull, it's a reconstruction. And this reconstruction was done by scanning the amber in a CT scan machine, which created thousands of images. Actually here was 1,860 images. And these images we have reconstructed here with a special computer program to make a three-dimensional picture of the skull that can be looked in all different directions. This is a major discovery of an animal group, and it wasn't so clear exactly why there's different anatomy was there until we found this species. This CT scan analysis needed to be expanded, so it had to go to the synchrotron in Australia for a second analysis with another type of X-ray beam generated by the synchrotron and higher resolution data. The total information of these CT scan procedures and then the high resolution scans in Australia at the synchrotron gave us a total information package that we could use to make a three-dimensional model of this skull using a 3D printer.
This is a piece of amber, and it contains unbelievable interesting information in it from an animal that last was seen, as far as we know, 5 million years ago. And we needed the world expert to find out what exactly it is. So we gave it to two of the world leading experts of it, and I give it first to Professor Evans. What he can tell us.
Thank you very much. So this little tiny piece of amber has got a skull inside it. And if you just look at it superficially, it looks rather like the skull of a lizard. But there's breaks and cracks in the amber, and so to get a better image of it, we put it in a CT scanner, and that will give us a 3D representation of what the skull looks like. And so when I was sent that 3D representation, I could see immediately that it wasn't a lizard, and in fact it was a kind of amphibian that is called an albinerpetontid. We call them albis for short, so we'll stick to albi because it's easier to say than albinerpetontid. And this is a group of amphibians that lasted from about 165 million years ago through till about 5 million years ago, perhaps a little bit more recently than that. But most of the remains that we have of it are disarticulated bones, individual bits of jaws, individual bits of skulls. Even those specimens that we have that are articulated, we have some from Spain, for example, they're very flat, they're two-dimensional. So this is the very first specimen that we've got that's completely 3D, three-dimensional. And so for the very, very first time, we can see the details of elements of the skull, of the brain case, of the jaw region, and how all these bits are put together. And that gives us a much clearer idea, or at least the basis of a clearer idea, to work out how they're related to modern amphibians. So they don't belong to any of the modern amphibian groups, they're related in some way to them, but they're really, really bizarre animals. And what this skull also helps us to do is to begin to put together some of the strange things that we knew about these animals. So we knew that they had a very strange jaw joint, but difficult to understand exactly why it was. And this three-dimensional skull from the amber helps us to piece together a lot of the puzzles that we had from isolated material. So it has a very strange jaw joint that kind of has a flexibility to it. It has a strange jaw joint at the back, which is a strange shape. And we knew that it had this long median process that ran underneath the skull that we thought might be connected in some way to a prehensile tongue mechanism, a ballistic tongue mechanism. And this great specimen has also got some soft tissue on it. So the soft tissue actually shows us the very tip of the tongue, and it proves that this long median skull element ends in the tongue And so this helps us to begin to understand why it's got this strange jaw joint that would have allowed the mouth probably to open better. This tongue would have been able to shoot out rather like a chameleon. And that's one of the really fascinating things about these little animals. Previous people thought they might have burrowed, but we always thought underground, but I always thought that that wasn't likely to be the case. If you have a ballistic tongue, it's no good being underground. You've got to be above ground, you've got to be climbing so that you can climb up on things. Finding one of these in amber shows us that they were related around, they were around tree stumps. So it shows they had to have been above ground. It brings all of these kind of ideas that we had about this group together in a really beautiful way. And so we have a much, much better idea not only of the skull structure, but of their behavior and what they might have been, how they might have been living.
The name, a little bit personal for me too. First, the name is Yaksha, a protector for the treasures under the tree. And then the second part is my name, honoring my long work in Burma and a discovery of this piece. That was decided by the scientific group. It's nice, it's very beautiful piece too, and it's the center point of the collection. It is in the Peretti Museum Foundation as a holotype that you call all the animals that have discovered the first time. You have a deposit of it in a museum, so it is always there because people need to go back and restudy something. This sample must be available for the public and safe forever. So we could guarantee that. It's in Switzerland in our collection, and as the centerpiece, most beautiful one, most spectacular one, and solving a problem what these Albaner Beton animals were doing, snatching with the tongue an insect, and that stunned the people, you know. So it's out there in the world now, this problem solved, the unbelievable centerpiece of our entire amber collection.
So it's a very, very important discovery. It changes what we know about life and how it evolved on Earth. It was inside that amber preserved for us to discover it. But the Peretti Museum Foundation have much more ambers in its collection, and this is just the beginning.
这不也没有，不也没有，冒充那干的。